Log on, tune in, find out. Another good idea from Cambridge.
Well, welcome to the third lecture in our Darwin College lecture series on the theme of life. This week we're moving to what you might call the fundamentals of animal life, what I suppose you might call the bioelectrical machinery that underlies it all, and the subject is the spark of life. And our speaker is Francis Ashcroft, who's the Royal Society Research Professor at Oxford University, based at the Physiology Laboratory. Professor Ashcroft got her first degree in Cambridge at Girton College, and then did her doctorate at Oxford, and accumulated a very impressive research achievement, establishing the link between blood sugar levels and insulin production, which, quite apart from the theoretical implications, enabled her with colleagues to identify the DNA sequencing for the relevant pathways for this, which in turn permitted the early identification of babies who are predisposed to diabetes, and that in turn led to greatly improved remedies for that particular illness. And now she's using her insights into the body's electrical controls to investigate influences on obesity. Among her many honours, Professor Ashcroft this year has been announced as the UNESCO winner of the UNESCO Women in Science Award for 2012. And she's also a renowned populariser of science, appearing in front of many audiences, having produced a very successful book called Life at Extremes, and having one coming out courtesy of Penguin Books in June called The Spark of Life, which is the title of the lecture. So it's a great delight to introduce Francis Ashcroft.
Thank you very much. Can everyone hear me? Good. So I'd like to begin by saying thank you for a wonderful introduction, and thank you for inviting me to come and speak tonight. And I'm going to begin by introducing you to my friend Ed Jackson, who runs a ranch in Oklahoma where he farms goats for meat. Now, Ed is a temporary sheriff, and he's quite a colourful character, but his goats are even more unusual. And that's because although they appear completely normal when you see them here, if they're startled or excited, as you can see here, then their muscles seize up and their legs become very stiff so that they find it difficult to walk properly. And sometimes, as you can see, they even fall over. And there are stories of the whole herd collapsing when the train goes past the field and sounds its whistle. And although it looks really rather dramatic, the goats aren't hurt by this experience, and they get up again just fine in a few minutes. And I can show you an even more interesting video, if it works, which is shown on the next slide. Come along. You see that sometimes it's much more dramatic than you see the first time. Well, as you can imagine, the goats have a pretty tough life because everybody likes to scare them to see them fall over. And the reason that they collapse is because they have an inherited disorder of muscle contraction. And this is because the electrical activity of their muscles is not normal. And although it's not widely appreciated, goats and humans and just about everything else, we are all electrical machines. And everything that we see and do, from you listening and hearing to me talk, to you thinking, to you feeling emotions, to moving your arms and legs about, is down to the electrical activity that's taking place in the nerve cells in your brain and the muscle cells in your limbs. And in turn, ultimately, that electrical activity is down to a set of little-known but extremely important proteins known as ion channels. And in this talk, what I'd like to do is give you a brief introduction to the history of animal electricity, then show you how ion channels work, then say something about the fact that they're the targets for many therapeutic drugs as well as many toxins, and that they play a very important role in human and animal disease. And finally, I'll end up telling you about some of my own work. This is supposed to be an ion channel. I'll just briefly mention that they are proteins that sit in the membrane of every cell in your body and act as little gated pores. And I'll come back to that later. But what I'm going to end up with is showing you how understanding how these ion channels has worked has led to patients like the ones shown here who were born with a rare genetic form of diabetes to be able to throw away their insulin needles and take a pill once or twice a day instead. So the science of animal electricity begins here with Luigi Galvani, who is known as the discoverer of animal electricity. And back in the 1790s, he, like many others, was very interested in the science of static electricity. And he used a machine like this one to generate electrical sparks. You simply turn the handle and the sparks arrive here, and my thing is running out, I hope, or not. And what he discovered was that he happened to have a set of freshly dissected frog's legs lying on his bench, and he noticed that whenever his assistant, his wife in fact, touched the metal implement to the nerve that supplies the frog's legs, the legs twitched wildly, but they only did so at the precise moment that a spark was generated by this electrical machine. So what Galvani surmised was that the electrical spark was stimulating the muscle to twitch. And his and he knew from the work of Benjamin Franklin that lightning is also a form of electricity, so he decided to see whether lightning could stimulate the legs to twitch. So he connected a nerve to this long metal wire and attached it to a metal spike on the top of his house, pointing up towards the sky, and he did indeed observe that when the lightning flashed, the frog's legs twitched. And being a careful scientist, he of course did the control experiment and looked to see whether the same thing happened on a day when there was no electricity, no lightning around. And this time what he did was he attached a brass hook to the nerve, the main spinal cord nerve, and he hung the legs over the balcony. So he didn't quite do the proper control experiment. And nothing happened when he did this. But then he got a bit impatient, so he fiddled around with the legs, and he discovered that occasionally, even when there was no lightning around, they did twitch. And he noticed that they did so whenever the brass hook touched the iron railing. And he interpreted his experiments to indicate that the muscles are not only stimulated by external electricity, but animal cells produce their own intrinsic electricity, and this is what's causing the muscles to twitch. However, the interpretation of his experiments was contested by his countryman, Alessandro Volta, who said, no, that's not right. What's going on is that an electrical current is created by the juxtaposition of the two dissimilar metals. And he went on to build the first ever electric battery. And here you see it. It's stacks of silver and zinc discs separated by a little salt solution, just common salt. And here you see one of his very first voltaic piles, as they were known. And if you really want to see one, you can go and see in the Royal Institution the one that Volta gave to Faraday. Anyway, despite the fact that there was this huge controversy about what caused the muscles to contract, Galvani was in fact right, and animals do produce their own electricity. And his experiment produced much excitement. And wherever frogs were available, scientists and laymen alike would go out and see what happened when they tried to make the frog's legs twitch. And they performed these experiments not only on recently dead animals, but also on recently dead humans. And they used people from the guillotine or people who'd been hanged in local prisons. And in fact, they brought them to London. This happened in London. And these spectacular theatrical demonstrations were carried out by Galvani's nephew Aldini as a kind of way of emphasizing the importance of his uncle's research. And here what you see is a voltaic pile. What Aldini did was to connect a conductor here between the voltaic pile to the nerves that supply the head. And this caused the most fearsome facial grimaces. And I can read from you here what they said. He said, the jaw began to quiver, the adjoining muscles were horribly contorted, and the left eye actually opened. And unfortunately, worse was to come, because in Edinburgh, what they did was they applied the electrical shock to the arm, the nerve stimulating the arm, whereupon the corpse raised its arm and shook it at the audience, creating great consternation because some people actually thought it had come to life. And this was probably what inspired Mary Shelley's famous novel Frankenstein. But it did actually provide some useful scientific information as well, and that is, there was then seen which nerves supplied which muscle. So if we skip forward to the present day, we now know that the muscle contracts because an electrical impulse is sent along the nerve that supplies it. And that electrical impulse is down to the activity of ion channels. And these, as I've said before, are tiny little pores that sit in the membrane of our cells. And what happens is every cell in our body is surrounded by a membrane that acts as a barrier between the inside of the cell and the outside environment. And this is made of lipids, of fats, and these are impermeable. So we need transport proteins that can shuttle things backwards and forwards across the membrane. And one of the things that they shuttle are charged ions, sodium, potassium, and so on. So here is a sodium channel, and you can see when it's shut, no ions can go through, and when it's open, sodium ions can pass through, many hundreds of thousands of them per second, and this, the sodium ions are high outside and low inside the cell, so they tend to move from outside the cell into the cell when the channel's open. And this is just a little simulation. Look down on top, showing the channel opening and closing. And it's the opening of these ion channels that underlies the nerve impulse. So put very simply, at rest, the sodium channels are shut. If we apply a stimulus, the sodium channels open and sodium ions rush into the cell. And because they're charged, they carry an electric current, which you can actually measure. And also what happens is the inside of the cell becomes more positive, as you can see, because there's more positively charged ions in here. And shortly afterwards, potassium channels open, and the potassium channel ions move in the opposite direction. So what you get, if you record this, this change in potential, is what you'll see is, to begin with, it's at rest, then the sodium channels open, carrying the inside of the membrane positive, and then the potassium channels open, returning it to basal again. And this is really pretty much what the nerve impulse is. So this is actually a Cambridge story. It was two Cambridge scientists, Alan Hodgkin and Andrew Huxley, who worked partly in the physiology department and partly down at Plymouth, who recorded the first action potential with an intracellular electrode and figured out how it worked. And of course, they won the Nobel Prize for this. And one thing that contributed to their success was the fact that they used the giant nerve fiber of the squid. And this is shown here. So this is a kind of trunk of nerve fibers, and this is the size of an ordinary nerve fiber here. It's really quite tiny compared to this giant axon, or nerve fiber, which is used by the squid to produce a very rapid contraction of the muscle, enabling it to have a jet-propelled escape response. But from the point of view of the physiologists, it's much more useful for something else, and that is recording action potentials or nerve impulses. And here you see Hodgkin and Huxley's photograph of the nerve fiber running down here. You can see how big it is. And into it, you can see they've inserted an electrode. And what they've done then is to measure the potential between the inside and the outside of the cell. And when they started, this is what they found, a resting potential of around minus 60 to minus 70 millivolts. And when they stimulated it, they got an action potential, which, as I've told you, we now know is due to the opening of first the sodium channels and then the potassium channels. And Hodgkin and Huxley, this is also an interesting story. They recorded this first action potential in 1939. And of course, they were incredibly excited about it. But the experiments had to be abandoned a few weeks later because Germany marched into Poland And the Second World War started, and they were needed for much more important things. And it wasn't until 6 years later that they were able to return to Plymouth to take up the experiments again. And of course, it took some time to get things going because the labs had been partly demolished in air raids, squid were in short supply, and as Hodgkin said, they'd forgotten much of the technique in the intervening years. Nevertheless, it didn't take them long. Within a few years, they've solved the problem of how nerves work. And most of the experiments were actually done within just a few short weeks in one summer. Now, today, what ion channelologists like myself are interested in understanding are these properties. What causes the channels to open and close? A property we call gating. What determines which ions go through them, whether it be sodium or potassium ions? Why are they important for the function of the cells? Every cell in our body has got these things, and everything from a redwood tree to a bacteria. So what do they do? And finally, if they go wrong, how do they cause disease? Now, we know that some of these, if we just take the first one, we know that some of these channels are opened by the change in the voltage across the membrane. So basically, they have a little voltage sensor, which when the channel is shut, is sort of interacting with the negative potential on the inside of the membrane. And then if the potential is altered, then it pops up to the other side, and that causes a conformational change that opens the channel. And some of them, like the ones that I'm passionate about, are opened and closing by binding a chemical ligand, as shown here. And I'm just going to show you what they actually look like, because they don't look like those cartoons I've shown. And in order to see what they look like in the last few years, the channels have gone to Stockholm all over again and won a few more Nobel Prizes for Rod McKinnon, who solved the atomic structure of the first channel. And what you see here is what it actually looks like. This channel is built up of 4 different subunits. Each of one is a different color. It's just twizzling around to give you an indication of what it looks like. This part up here is in the membrane, the bit at the top. And here you see, in a moment, this is the channel opening. And here comes in the chemical ligand that's causing it to shut. So, and this is what the eye view of the ion might be. You see, there's quite a big hole for it to go through when it's open. So, what else do they do? Well, one of the really important things, I think, about ion channels is that they're a target for many toxins and many therapeutic drugs. And during his voyage in the South Seas in 1774, Captain Cook wrote about the fact that he came home one night and he found a bit of fish. They ate a little bit of this fish, and they went to bed. And then at about 3 or 4 o'clock in the morning, they found themselves seized with an extraordinary weakness and numbness all over our limbs. I'd almost lost the sense of feeling, nor could I distinguish between light and heavy bodies of such as I had strength to move. Fortunately, they vomited up most of what they'd eaten, because in the morning, one of the pigs which had eaten the same thing that they'd eaten was found to be dead. And when the natives came on board, they emphasized that these were not fish that were supposed to be eaten. And it's very likely that Cook had eaten some of the pufferfish, which, as many of you may know, the liver, intestines, and ovaries of this fish contain a virulent neurotoxin known as tetrodotoxin. And in Japan, the pufferfish is known as fugu, and it's considered a great delicacy. But it's an expensive delicacy in more ways than one, because if you eat the wrong bit of the fish, I mean, the idea is to eat it, of course. If you eat the wrong bit of the fish, then, of course, it can kill you. And every year, despite the fact that fugu chefs are licensed, around 50 people die of tetrodotoxin poisoning, usually fishermen who've inadvertently eaten their own catch. And this is a Shinto ceremony to celebrate the pufferfish, which you see here. Now, what TTX does is it sits in the outer mouth of the sodium channel and just physically plugs it up. So the sodium ions can't go through, so what that means is the channel doesn't work, and what that means is that nerve and muscle impulses are inhibited, and the victim dies of respiratory failure. You can keep them alive if you immediately put them on some sort of artificial ventilator until the poison is washed out of their system. I always think it's rather amusing that somebody else very famous has been poisoned on 2 separate occasions by pufferfish toxin, and that is 007. But both in Dr. No and in Octopussy. But as you can imagine, he emerges from these experiences shaken but not stirred. I couldn't resist. And there's a vast array of poisons from spiders and shellfish and, in fact, all sorts of marine creatures, scorpions as well, that interact with ion channels. And some of them act extremely quickly. And I'm just going to show you a video of one of these. So this is a cone snail. And in this little proboscis here, it has a dart that it uses to inject its prey. And what you see here is how rapidly this can occur. So here it is with its little proboscis coming out. It's just about to inject Captain Nemo here. And you see the fish is now completely paralyzed and is being slurped up for lunch. And I'm afraid that there's enough toxin in these cone snails to kill a person as well. But ion channels are not just targets for all sorts of toxins, they're also targets for important therapeutic drugs, and local anesthetics work by blocking sodium channels. So next time you go to the dentist, you can remember that this is what's happening to the nerve fibers. They're being inhibited because the sodium channels are blocked. And this is the side view of the sodium channel, which is quite exciting because we've only just got its structure. It came at the end of last year. And you can see here in cross-section, in side view, so I don't seem to have a pointer working here. Is there any possibility of another pointer? And anyway, what you can see is here comes in the side of so the local anesthetics come in through a little window in the side of the channel in the middle of the membrane. So they dissolve in the membrane and move in through here. And you can see they have access right into the center of the paw. So ion channels are also very important in human disease. And this is an example of what happens in some patients who have a mutation in their muscle sodium channel. And I want you to watch very carefully what happens to the hand, which is in this bucket of cold water. So what he's been asked to do is to clench his fists and then open his fists. And you'll see, sorry, the quality of the video is not so great. Now, have a look and see. So the hand that was in the cold water, it's very difficult for it to reopen. And this is because he has a mutation in the sodium channel, which means that when the sodium channel is cooled, when it's colder, it starts to become leaky to sodium ions. And the consequence is that that means the muscles are excited more, and so the hand cannot relax. And this might remind you of what we saw in the first slide, the disease known as myotonia congenita, which is not only found in goats, but also in humans. And in fact, it was first described by the Danish Dr. Thomsen in himself and his family. And he kept quiet about it until he was about 60 years old, when his son was accused of malingering and not joining the army, his young son. And so he published his studies to save his son. And it was the goats. So what happens with these people is that they find it very difficult to let go when their muscles have been undergoing a strong contraction. So if you can imagine coming and picking up a very heavy suitcase, they won't be able to let go of the suitcase when they try to. And there's a wonderful story told to me by one patient who wanted to run a race, and he was crouched at the starting line like this, and just like the goats, when the gun went off and he tried to run very fast, everything locked up. And he says that, although he didn't fall over, he felt like he was running on stilts, and he didn't really start running until the last few meters of the race. And people with this disease tend to have very well-developed muscles, as you see here, because in addition to the problem of over-contracting when startled, they also are undergoing tiny little contractions all the time. It's a little bit like doing isometric exercise, and which is why they have this very fine physique. Just unfortunate the muscles don't work very well. And the goats turned out to be the key to understanding what this disease was all about. And this is Shirley Bryant, an extraordinary, wonderful, flamboyant character from Cincinnati who, even in his 70s, would wear his hair in a ponytail and go disco dancing. And he read about these goats in about 1963 or something, in the 1960s anyway, serendipitously when he was just browsing through the veterinary record, as one does, and he heard of the goats falling over when the train went past the field, and he thought, that sounds like the human disease, so he decided to investigate. He got some of the goats, and he took a small sample of muscle from between the intercostal muscles here, which doesn't hurt, and is also done on human patients quite often. And what you see here is what happens when he stimulates a normal muscle fiber or a muscle fiber from the myotonic goat. And in the normal muscle, you just elicit one muscle impulse, one action potential. But in the myotonic muscle, you get many of them, and they continue even after the stimulus has stopped. This is a stimulus here. So what's happening is the muscle is being excited more, and that's why it's contracting more. And actually, just make sure I have to tell you that this also has a Cambridge connection. And some of you may have spotted it down here. Because Shirley was pretty convinced that what was going wrong here was this was a problem with a muscle chloride channel. And he wanted to measure the chloride current that flowed through this channel when it was open, but he didn't have the right equipment to do so. The person who did, had just developed it, was a man called Richard Adrian, who worked in the physiology department here in Cambridge. And so Shirley decided in the 1970s that he would come to England with 4 of his goats in order to do experiments with Richard Adrian. But it wasn't very easy to import the goats into England because of a disease known as blue tongue, and a special parliamentary order known as Goat Importation Act No. 1 had to be passed to allow their importation. And despite all this careful preparation, Shirley arrived in Heathrow waiting for the goats to come, and his collaborator was supposed to send the goats, and he forgot to send the papers with him. So the goats arrived at Heathrow, no papers, and were in imminent danger of being shot. But fortunately, Richard Adrian's father, Lord Adrian, and Master of Trinity, was able to sort things out. So ion channels aren't only confined to the skeletal muscle that moves our arms and legs. They can also occur due to mutations in the cardiac channels that are responsible for the electrical activity of your heart. And mutations in several different genes give rise to a rather horrible disorder known as long QT syndrome, which is characterized by an abrupt loss of consciousness and sudden death. And it's particularly tragic because it affects very young people. This is one young man whose story I found on the web, and this is even more tragic because Jimmy here collapsed when he was playing football in his early 20s, and 2 years later his sister also collapsed and died of the same disorder. And what happens is they have a mutation in a potassium channel in the heart, and because the potassium channel doesn't work, what happens is, I already told you the potassium channel's open to restore the potential of the nerve impulse. Well, they also open to restore the potential of the cardiac action potential, cardiac impulse, which you see here. So if they don't open very much, or they're not working very well, you get a much longer duration impulse. And the consequence of that is it's reflected in the electrocardiogram. The electrocardiogram is nothing more than a representation of the electrical signals that are going on in the heart, and these can be picked up from surface electrodes on your chest. This is a QRS wave, which indicates the beginning of the cardiac impulse, and this is a T wave, which indicates the end. So if you have a longer duration action potential or impulse, electrical impulse, you will have a longer distance between the Q and the T waves of your electrocardiogram, and that's why the disease is called long QT. And I'm just going to show you what happens I think I've missed a slide. Yes. So this I got from Dennis Noble. And what you see here are the two lower chambers of the heart, the ventricles. And when they are red, they're relaxed, so the heart is filling. And then what happens is the impulse comes along and takes the potential more positive, and that's blue. So now the heart is resting. And what you should notice about that, if we see it again, is that everything is contracting synchronously. So the electrical stimulation and the contraction is synchronous, so the heart can beat nicely as a pump. And if you have one of these mutations, this is what it looks like. It's electrical mayhem. And the whole of the heart is then quivering, as Vesalius said, like a little bag of worms. And this cardiac arrhythmia means that, of course, you can't pump the heart properly, so this is fatal. And this is why the patients are susceptible to arrhythmia and die. And you can get this disease not only from inheriting a genetic mutation, you can also get it from many drugs. And some years ago, it was discovered that a very common drug known as tafenidine, which used to be an over-the-counter treatment for allergy, many of you have probably taken it, I certainly have, actually had a side effect of blocking this potassium channel in the heart. And it caused a slight increase in the risk of sudden cardiac death. So it's been removed from the market. And many other drugs were subsequently shown to have effects on this ion channel. So nowadays, every drug has to be screened for its effect on these ion channels in early stage development, because many drugs fail as a consequence. It's very expensive, this channel. So, the other thing is, I mentioned earlier that ion channels are not just in nerve and muscle fibers, they're in just about every cell of the body, and they govern our lives right from the moment of conception until that of death. And, in fact, even before conception, because it turns out that an ion channel in sperm is critical for its ability to win the great sperm race. And that's why you and I are here, because of those ion channels. And what happens with a sperm is when they're ejaculated, from the moment of ejaculation, they must fight all the way to the egg by lashing their tails. And this is an example of normal swimming. You know, it's just wriggling along up the female reproductive tract. And when they get near the egg, they get more excited because the egg secretes progesterone, and it switches to this very massive whip-like motion. And this is essential because the sperm needs to have a pretty strong thrust to be able to penetrate the membranes that surround the egg. And if it doesn't have that, it can't fertilize the egg. And it was this chap here, David Clapham, who discovered these ion channels, which are found in this particular tail of the tail of the sperm. And you can see them here where they've been stained with a red dye. So these are the Catsper channels, they're known as. And this is, again, what happens. The normal ones, when they're hyperactivated with progesterone, they beat like this. The ones in the mice which don't have, where they've been knocked out, so this animal doesn't have any Catsper channels, they don't work properly. And the consequence is that every single one of these mice is infertile. And in fact, this is the most complex ion channel in the genome because it has at least 4 different subunits, and it's got many accessory ones as well. And it's also different ion channels are also involved in plant fertilization. I don't know if you know, but here is the pollen grain extending a pollen tube. It has to grow down the whole of the long female apparatus down towards the egg, and it carries the sperm with it. And when it gets there, what happens is the cells that surround the female egg release a chemical known as this strange name, ZMES4, which diffuses to the end of the pollen tube and opens up an ion channel there and causes it to swell up and burst and liberate the sperm so that they can then fertilize the egg. And we also use ion channels as lethal weapons. This is an example. So what happens is this is a T lymphocyte killing another cell, another blood cell that's infected with bacteria. And what happens is they secrete giant channels that go and plug themselves into the membrane of the target cell and form a huge great hole like this, which allows loads of the important constituents of the cell cytoplasm to be released from the cell, and the cell simply bursts and dies. And this one, Staphylococcus aureus alpha toxin, is particularly nasty because it causes blood poisoning if it gets into your red blood cells. So I wonder if you've wondered why chili peppers taste hot. Well, it's because they contain a chemical known as capsaicin, which works on ion channels in your mouth. And this is Wilbur Scoville, who calibrated the potency of chili peppers by diluting them and then seeing how much effect they had when placed on the tip of the tongue. And you see, if you have just a very green bell pepper, there's really no Scoville units. But as you go hotter and hotter and hotter, you get many, many more of them, right up to the US grade pepper spray, which is used in warfare and, unfortunately, for controlling citizens. And it was David Julius who showed that the capsaicin receptor is actually an ion channel. It's here, and it's not only sensitive to chili pepper, to capsaicin, it's also sensitive to noxious heat. And in fact, it turns out now that there's a whole range of these channels, each of which is sensitive to a different thermal scale, right down here from the ones which are sensitive to cold and also to mint and eucalyptus, which is why they taste cool, up to here with the ones that are affected by heat. And even more interesting, I think, is this. This is a pig trying to go to sleep, and here are the vampire bats coming along, attracted to it by the heat of its body. And if you look when it gets there, this is a truly disgusting thing, which I think is rather awful. Here they are lapping up the pig's blood, all seen in a thermal imaging camera. And the reason that the vampire bats get there is because they have TRPV1 channels which are able to sense body heat. And they're localized in these little organs here known as leaf pits, which have nerve endings, and in the very end of the nerve endings are these ion channels which sense temperatures of about 30 degrees. And they're also ion channels, as you see, are definitely the meaning of life. And here they are at the back of the eye in the retina. And in the retina, we have photosensitive cells. And in the photosensitive cells come in two types, the rods, which are sensitive to very low light intensities and enable us to see in moonlight, and the cones, which are sensitive to color and are also responsible for visual acuity. And ion channels are very important for both of these types of cells. And in particular, I'll just show you one example here. So these are the cones used for color vision. What you see here is an ion channel which is open all the time in the dark. And when a light comes along, it interacts with a photosensitive chemical called opsin, which triggers a cascade of events which results in the closure of these channels. And what happens in some patients is that they have mutations in the channel here, which mean that this closure never happens. So the channel's always open, so it never detects any light. And the consequence is that this is what they see if they look at this scene here. This is what, if you're not colorblind, this is what you and I will see. If you have no cone vision at all, this is what you'll see. Not only is it black and white, you're also photophobic, you're very sensitive to bright light, and everything is out of focus because the cones sit where the light rays are focused. So these are the people who Oliver Sacks writes about in his wonderful book, The Island of the Colorblind. So now I've given you a kind of flavor of what ion channels do and why they're important. I'm going to tell you a little bit about my own studies and why I'm passionate about this particular ion channel that's known as the K-ATP channel. And here it is. We don't have a crystal structure for it yet. We're struggling hard to get it. But understanding how this channel works has made a difference to young children like here, who was born with diabetes and ended up in hospital a few weeks later in diabetic ketoacidosis. So I have to give you a little bit of background. So what you probably know is that it's very important that your blood sugar level is controlled within very narrow limits. If it drops too low, you only need this to happen for just a few minutes, and your brain will be starved of fuel, and you will suffer brain damage. If it goes too high, you'll get all the complications that happen in diabetes. So insulin is very important in controlling your blood sugar level. So after you've had a chocolate biscuit or something, your blood sugar level will go up, your glucose will go up, and insulin will be released from the beta cells of the pancreas. And this will stimulate the uptake of glucose into muscle, liver, and fat, and so return your blood sugar level to its resting concentration. It's a beautiful example of homeostasis. And if insulin action in the tissues is impaired, as happens if you get too fat, then your blood glucose will go up because insulin is not as effective. And the same thing happens if you don't secrete enough insulin. And this is what I'm interested in, what regulates the secretion of insulin. Well, you may say, well, this is all very well. Why don't people just take insulin? You know, we've had insulin since 1920, so 1922. What's the problem? Well, the problem is that insulin is not a cure. This young man here looks very fit and healthy, but you'll notice he's got a little hospital armband on here. And this is because he's just had a heart attack, and as you can see, is really young. And diabetes leads to an increased risk of heart disease, increased risk of blindness. It's one of the commonest causes of blindness in the population. Poor circulation, which leads to tissue damage. It's the commonest cause of non-traumatic amputation. Also causes loss of sensation in the extremities due to nerve damage, and about 30% of the people on dialysis are there because of diabetes. I want to emphasize that the risk of complications is reduced by good control of blood sugar levels, as any of you who are diabetes will know. But I should also mention that there are several different types of diabetes, and this is sort of important. So type 1 diabetes is what's often known as juvenile diabetes. It occurs in childhood, and the beta cells are destroyed by an autoimmune attack. So it's kind of an example of friendly fire. They're killed off by the body's own cells. And this means that people with type 1 diabetes must be on insulin for the whole of their lives. Then there's type 2 diabetes, which, looking at the size of the audience, many of us will have it or are going to get it because it's affecting about 5% or more of the population, and it's currently on the rise. It's very high in the UK, and it's massive throughout the world. And although it affects adults, increasingly it's affecting kids, and that's because as a population we'll all be getting more obese. And here the beta cells are present, but they're just glucose blind. They don't respond to it. But multiple genes are involved, and it's very difficult to sort out what goes wrong. It's much easier if you take neonatal diabetes, which is a very rare genetic condition where just a single gene is mutated. And we now know, as I'll show you, that 50% of patients who have diabetes at birth are there because of K-ATP channel mutations. So, when I first came to Oxford many years ago, back in 1984, I set myself the following question. How does a rise in blood sugar stimulate insulin secretion, and what goes wrong in diabetes? And this is what we knew. We knew that if you have a rise in blood glucose, the glucose is taken up into your beta cells, it's broken down to produce something that stimulates electrical activity in the cells, and that leads to insulin release. And I had a kind of idea that an ion channel might be involved, as you might guess. And what was wonderful was that just at that time, a new technique had been invented that allowed me to look at the activity of an ion channel. This was invented by Erwin Neher and Bert Sackman, as you can see here. And they won the Nobel Prize, here's Sackman, for this. And this technique was published in 1981, and I got to Oxford in about 1982 to 3, so I struggled away with this method. And this is what I did. I brought a glass tube down onto the surface of a single beta cell so that I could isolate just one ion channel in the patch of membrane under the electrode, and that allowed me to record the very tiny currents that flow through the channel when it's open. And because I wanted to know what glucose did, and I knew that it had to be broken down, I added glucose out here to the bath solution so that it had to come into the cell, be broken down, and then work on this channel. And so this is the sort of thing I was looking for. This is what single channels look like. Here they are with a little downward deflections, this one channel opening, and when it's double out, it's two channels opening, and so on. And this is the sort of thing I used to dream of for night after night after night. And this is what I found. One day, about 8 o'clock at night, all on my own in the lab. And so here I had, when there was no glucose around, and the cell was just sitting there, there was this little ion channel popping away very happily. And then I added glucose, and as you can see, it's quite obvious, it vanishes. So I thought at the time, oh, shit. The experiment's not working, you know. Something's gone wrong. And then I thought, well, maybe not. Maybe I'll just take the glucose away again and see what happens. And it came back. And so you can imagine, I was ecstatic. And you can see I'm still excited by this, even all these years later. I mean, this for me was my eureka moment. Oh, well, one of them. And then, of course, the question was, so glucose closes this channel. And it does so because it's broken down. So what is the breakdown product of glucose? And it turns out that this breakdown product of glucose is a chemical known as ATP, which is why this channel is known as the K-ATP channel. And I had these experiments all ready to go to the journal when I was sent this paper by these guys to review. So I never got that published. And then I thought, well, OK, I'll put this together with the effects of glucose and show that the same channel is blocked by glucose and is blocked by ATP. But this chap, I got a paper to review from him. And I thought, And anyway, it was very nice. The paper was accepted. My data never saw the light of day. And then a year later, I met Patrick in a swimming pool in Alicante, as one does. And we decided that it would be much more fun to collaborate than compete. And we've been collaborating ever since. And now he's even in Oxford. He's Swedish. He was in Sweden at the time. So I see I've got to talk more rapidly. Anyway, so now this is what we have. We have a channel which is, when it's open, it allows potassium ions to go through. And when it binds this chemical called ATP, it shuts and no ions can go through. So by 1985, we knew that when metabolism was low and ATP was low, this channel was open. And that prevented insulin release. And when metabolism went up, ATP was generated, shut the channel, stimulated electrical activity, and insulin was released. So the important thing is that when the channels are open, no insulin is released. When the channels are shut, insulin is released. And it doesn't take a genius to realize that if the channel is insensitive to ATP, it will never shut in response to ATP. And so even when blood glucose levels are very high, no insulin will be released, and the result will be diabetes. So way back in that night in 1984, I'd already figured this out when I saw the experiment, and I thought, this is what causes diabetes. The problem was, it took 20 years to show it, because first we had to get the gene sequence for this protein, and there's two of them together that make the channel, so that wasn't easy. And then we had to find the patients. And once we'd sequenced the gene and we could screen human patients for mutations associated with diabetes, we started by looking at type 2 diabetics who have diabetes late in life. And I thought even at the time, we shouldn't be looking at these people. We should be looking at people who are born with diabetes. And I said to my medical friends, well, what about the ones who are born with it? And they said, oh, they don't exist, Fran, they don't exist. But fortunately, one man knew better, and that's my colleague Andrew Hattersley down in Exeter. And he instituted, they're very rare, these people, and he instituted a worldwide search to collect DNA from these patients. And then he screened them, and the first gene he chose to study was our channel. And he found, I've never forgotten the time, he called me up on the phone. He said, Fran, I've got something to tell you. I think you should sit down. And he told me that he'd found the first mutation and asked us to study it. So this is a very rare disease. It occurs in about 1 in 200,000 births, and it's characterized by very high blood sugar levels within the first 6 months of life, and usually a low birth weight because the patients haven't had any insulin. And we know that in some cases, it doesn't just cause neonatal diabetes, it also causes neurological problems. The children walk and talk very late, they have floppy muscles, and in 3% of them, they also have epilepsy. And poor Cameron has the developmental problems. And we've found millions of mutations in the channel. I'm just going to show you. Here they are. You'll see anything that's red here is associated with diabetes, and they're all in bits of the channel that move when it opens and closes, or in this region here, which binds ATP, which is this purple molecule. And what we've been able to show is, whereas ATP will close the wild-type channels, it doesn't close the normal channels, it doesn't close the mutant channels. So basically, this is what we have. We have channels which are ATP insensitive, so they remain open, so no electrical activity, no insulin secretion. But the marvellous thing is that this has had implications for therapy, because these people had been treated all their lives by insulin injection, because it was assumed that they had an unusually early form of type 1 diabetes, in which I previously told you all the beta cells are destroyed, and you have to have insulin. And not everybody feels happy, especially if they're tiny, about injecting themselves with insulin. And this is also very difficult if you've got a 6-month-old baby. But what we knew, because of work that we've done years before, is that there's a drug known as sulfonylureas, which is used to treat people with type 2 diabetes, which had been used for more than 50 years to treat adult patients. So it's very safe, no side effects, apart from hypoglycemia, if you take too much. And it was in routine clinical use, so it could be immediately trialed in patients with neonatal diabetes. And the transfer procedure was set up by Andrew, and more than 60 patients have now transferred to drug therapy, and it's had a massive effect on their clinical condition and their way of life. And what you see here is an index of the average blood sugar concentration for the previous 6 weeks, and when they were on insulin and after they transferred to sulfonylureas. And you see that the value of this index here, HbA1c, comes down, which means that their blood sugar level is much better controlled. And really importantly and satisfactorily is that many of them are now in the normal range. So that means that their risk of getting all of those diabetic complications, which I discussed previously, is really gone. And it's, of course, very nice for the patients. They don't have to inject themselves with insulin anymore. More importantly, they rarely suffer from hypoglycemic attacks, which is a common problem if you take too much insulin. They never end up in hospital. And the parents now allow their little kids to do all sorts of things. And one of the patients told me that when she was younger, she had had a she'd had an episode of diabetic ketoacidosis, which had meant that she'd ended up in hospital in Thailand. So after that, she had never gone abroad in her life. She was 48 years old. She transferred to sulfonylureas, and now she's having foreign holidays every year. And another guy said to me, well, the marvelous thing is that I can now run a marathon. And I think for me, one of the nice things was this young girl who turned to me and she said, thanks to you, I can wear a dress. And I said, what do you mean? And she said, well, I've always had to have, a waistband, a skirt or trousers to hang my insulin pump from. And now I can wear a figure-hugging dress. So I think, for me, this has been such a wonderful thing, because as a scientist, I do science out of just sheer curiosity. I want to understand how it works. It's a fascinating puzzle that you want to try to sort out. But if you work in the medical, kind of related medical area like I do, there's always in the back of your mind, there's this hope that maybe something you do will actually have some benefit to patients one day. But you always imagine it's going to be long after you're dead. And it's an enormous privilege that, in fact, it happened in my lifetime. And it's been even more of a privilege, or an extremely humbling experience, to meet the patients who have this disease. We held a wonderful conference for them, a families' day at the Royal Society a couple of years ago. And now, what about the neurological problems? Well, I'm not going to tell you very much about that, but all of these things do happen, and this is just a little video of Ewell, and you can see at 2 years old, he really is not able to walk. He's a charming little boy, but this is the best he can do because he hasn't got enough strength in his arms and legs to stand up. Now, what we know is, and I'm not going to show you the video, we know that in some of the patients like Ewell, that if they get the drug, within a few weeks they are walking. But there are so few of them, and that really this is not a proper experiment. But I can tell you that those patients who, the parents of the patients who have suddenly started to walk after taking the drug, won't let us do the experiment of taking them off the drug. And but for me, then their brain problems, you know, their ability to read and write and these other things are still impaired. But what we're really interested to know is what happens in those children who are treated from birth. And she was treated from birth, and she's now 4 years old, and her development is so far normal. There are a few minor things, but you could get that in any child. And he didn't get the drug till he was 4 years old, and he is still rather impaired. He had it from birth. She has the same mutation, and she is meeting all her developmental milestones. So we're just keeping our fingers crossed and hoping that this will work. The real problem is how much of this drug we can get across the blood-brain barrier to affect their neurons. So just briefly, this is a story which is not lost in translation. Many mutant channels can be closed by the drugs, and more than 90% of patients have now transferred to drug therapy. And in some cases, their neurological symptoms also improve. So I'd like to emphasize that this type of work requires a very, very long time and a hell of a lot of money. As you can see, it's taken 20 to 25 years to get from finding the channel to a new therapy. And no matter how fast it appears in the final stages, there's a huge amount of work that goes on beforehand. And of course, I just flash up all the team who are currently working with me on these problems, and all my many collaborators, all the clinicians from across the world who sent us their mutations to look at, and in particular, my wonderful friend with whom this has been a great journey together. And I hope that I have convinced you that ion channels are in fact the spark of life. Thank you.
Well, back in the dim and distant past, when I got interested in industrial relations, there was a union called the Electrical Trades Union, which used to boast, not without reason, that its members could close down any workplace of any size in the entire country. Well, technology and economics have changed all that. They're no longer indispensable as they were. But clearly that's not the case with ion channels. They really are indispensable. They can shut down, as little electricians, they can shut down absolutely anything. What a lovely story of discovery, albeit frustrated discovery. And in terms of human happiness and human suffering, how very, very useful. That was a lovely talk. Thank you so much.